recording. Good. So, welcome to the PSI Subspace Virtual Talks. This is co-sponsored by the Center for Translational Research in Neuroimaging and Data Science, the Trend Center here in Atlanta, Georgia, and also sponsored by the IEEE Signal Processing Society. I'm Roger Silva, a research scientist here at Trends, and today it is my privilege to introduce our speaker, Dr. Benjamin Risk. He's an assistant professor in the Department of Biostatistics and Bioinformatics at Emory University here in Atlanta, Georgia. And he completed his PhD in statistics at Cornell University in 2015 and was a postdoctoral associate with the Statistical and Applied Mathematical Sciences Institute and the University of North Carolina, Chapel Hill, until 2017. Dr. Risk research focuses on neuroimaging and aims to further scientific understanding and medical research by developing, improving, and disseminating statistical methodology. His research includes dimension reduction methods, multimodal data integration, and the statistical impacts of MRI acquisition methods. Ben is an accomplished researcher working on very important problems in our field, with a focus on improving methods to analyze and integrate multiple data sets. Today, he will be presenting on data integration in neuroimaging studies. Dr. Risk, it is an honor to have you here today. Thank you so much for sharing your time with us. You have the floor.
Thanks, Rogers. Thank you for that introduction, and thanks for the invitation to speak with you all. So the slides I have posted on our GitHub, and the Brisk Lab, there's a repository called Presentations. I can share that link also later on. Let's see. Okay, so it's a little different. The click is working. Okay. So today I'll be giving a little bit of an overview on data integration, and then talking about two different approaches that we've been investigating. The first is a method that we call probabilistic joint and individual variation explained. And then in the second part, I'll talk about simultaneous non-Gaussian component analysis for data integration in neuroimaging. So just briefly, the overall goal here is to find information shared by multiple data sets that's collected on the same participants. So this general field of research has a few different names. So we're talking about multimodal data, particularly in imaging. We're talking about different imaging types. It's also called multi-view data or multi-block data. So you see some of these methods emerging in the genetic literature. And the idea that we're going to be exploring here is capturing that shared information using subject scores. So these subject scores are a way to capture the subject's phenotype from these like high-dimensional complex data. It's a way to achieve dimension reduction to get in a reduced subspace that's more interpretable. And so these subject scores can be investigated as possible biomarkers. So there's some papers related to the Trends group that have looked into this. And in part one, we'll be talking about integrating some features from T1 images with some behavioral clinical data. And our specific application will be looking at brain morphometry and cognition from the ADNI data set. And then in part two, we'll be integrating task fMRI with resting state fMRI, but more generally, the part two methods can be applied to multimodality. Say if you're looking at resting state fMRI and diffusion weighted MRI, etc. Okay, so this first part is joint work with my former PhD student, Dr. Raphael Murden, who's now an assistant professor in the Department of Biostatistics at Emory. And it's also work with my current grad student, Gavin Tien, and with Decheng Chu. So Decheng is in the Department of Radiology at Emory, and then also in the Department of Biomedical Engineering at Tech in Emory. So just a little bit of a background on this method called joint and individual variation explained. So this is a method that was proposed back in like 2013. And then there's a few different versions that are out there now. So there's another one from 2018. And the idea here is to define the shared information in the subject score subspaces. So we generally have these huge data sets that are n by pk, or they're not always huge. Actually, in this application, they're not necessarily huge, but so we have an n by pk data set. So n is going to be the number of subjects, and p sub k is going to be the number of features. And the idea is that we're going to be finding. Are you able to see my cursor when I move that around?
Yes, I can see it.
Okay, cool. I don't see I'm not a WebEx aficionado, so I don't see an actual high level, but hopefully that'll be good enough. Okay, so we have this data set. It's going to be n by pk, and x sub k, the kth data set, we're going to decompose into a matrix that captures the joint structure, and then a matrix that captures the individual structure. So I'll be using this word individual. It's a little confusing because it means unique to a data set. It's individual to a data set. It's not like a subject. So that'll be different. The subjects here are arranged by rows. So each row is a different subject. Okay, so this is the individual information, and then this is going to be the noise. The idea is that JK plus AK, that's the signal. So that's the information of interest, and that's going to be low rank. And then the way this jive model is defined is equating the column spaces of JK for different data sets. Okay, so this is a group about subspaces. So this is looking at linear combinations of the columns of JK as that. So that subspace is going to be the same across these different data sets. And then we're getting also the jive model also assumes that the column space of JK is orthogonal to the column space of the individual subspace for the kth data set. And the intersection of all those AKs is going to be equal to 0. And then we also assume that these noise matrices are isotropic in the sense that their singular values are approximately equal. Okay, so basically all the correlation and all the stuff going on is going to be captured in the JK and AK matrices. So that we're going to gain some insight into that as we kind of move through what those things mean. Ooh. Okay, the figure doesn't show, so I'll have to jump back and forth. Interesting. It's something on my computer. So when I was in full screen mode, that didn't pop up, but I think you can all see it now. Okay, so here's our.
Yes, I can see it now.
Okay, so let's just think about the case of two data sets, X1 and X2. And it turns out that this angle-based jive that I was just talking about in terms of the equality of subspaces has a close relationship to canonical correlation analysis. And really it comes down to doing a separate PCA on each data set to find that signal, that JK plus AK, which I called BK in that previous slide. And then you take these left singular vectors here. This is n by R1, this is n by R2. And then you actually find the canonical variables of those principal component scores. Okay, so it's also related to when you generalize this to multiple data sets to MCCA. But here we're going to take the two canonical variables and you basically average them and rescale them. Or you do average and rescale them. And that gives you the joint scores. So this is the subspace that's capturing that shared information. The way it's presented in the AGI paper is a little different, but I find this way a little more intuitive. So you can think of it as just doing CCA on the PC scores and then averaging those canonical variables. And then there's a step afterwards that estimates the individual signals by doing a subtraction of that signal matrix. You subtract out the joint signal to get back to the individual signal. But it's a multi-step procedure. Okay, we'll try to launch full screen again. Okay, so this one popped up. So I just kind of want to get a high-level view of the kinds of things that we can do with Jive. So this is from a 2017 Neuroimage paper. I was using this AGIVE algorithm. And the idea here is the data input were behavioral variables from the Human Connectome project. So the behavioral variables that we input included measures of task performance during a working memory task. So that's what the blue and the cyan are. Those are variables collected during the task, and then some of these other variables are related to cognition and includes like fluid intelligence and some of the other ones that you see. Okay, so then that data matrix is like 1,000 by something like 200 or something. I can't recall what the exact number is of the behavioral variables. And then the imaging data that we use are actually the pre-processed z-statistic maps for each subject. So we're not using any of the time information in this data set. We actually don't use time at all in this talk, come to think of it. We don't do it in the second part either. So here, the input data matrix for the image data set is going to be the vectorized z-statistic maps. Is going to be a row, and then we have each row corresponds to a different subject. So you can do a separate SVD, i.e. a PCA, on each of these data sets with appropriate standardization, etc. And just the general idea is that if you do this SVD and look at the first component, and you're just looking at the working memory map, so this is the two-back, the z-statistic is a two-back versus zero-back contrast, an estimate of the working memory. We see these areas in the prefrontal cortex pop out in some other areas, in the frontal cortex and the medians, and but we also see kind of this background information. And then when we do the joint analysis, when we actually try to find out what part of this variance is also related to these behavioral variables, which include task performance, we see that those working memory related regions tend to pop out nicely. And then if we look at what happens in the individual information, we see in the individual information, most of that, the behavioral data, this is task performance, most of that doesn't load onto the individual components because it got captured in the joint components. And then on the imaging side, we just see kind of this background level that it doesn't seem to have a lot of biological meaning because most of it was captured in the joint structure. Okay, so that was with the AGIVE version, and so I wonder if this is true for this group, but I argue that defining the joint individual structure in terms of subspaces can be a bit challenging to understand. So what does it mean for a column space of JK to be equal to column space of JK prime? So I like to have a generative model when I'm looking at these kinds of problems. So we're going to propose a probabilistic model that may improve interpretation. And then in addition to that, this AGIVE algorithm that I just kind of high level described to you, you estimate the joint components and then you do a basis subtraction to get the individual components. So it's really a two-step approach. And it may be that a simultaneous framework, if we can define a likelihood and simultaneously estimate the joint individual components, it could be that improves accuracy, not only in the individual components, but also potentially in the joint components as well. So I want to review a bit about probabilistic PCA. So in the PCA world, this is a relatively new way to think about PCA, and it's just a latent variable formulation of PCA. So you have X sub I, so I is going to be the ith subject, I is equal to 1 to n, and that's going to be contained in RP. So P would be the number of features, i.e. the number of variables. So I'm just going to describe this probabilistic PCA, which is just for a single data set, and then we're going to, our contribution is going to be extending that to multiple data sets. So X sub I is going to be equal to some mean. So generally we do pre-processing and don't worry about the mean mu. We just subtract it out. And then we're going to have these latent components ZI and this loading matrix. So these I call subject scores. Okay, so if you had an R-dimensional PCA, then Z sub I, you'd get R scores for each subject. If you had a rank R decomposition there. And then we have this isotropic noise. So that isotropic noise, under that assumption, it turns out that the solution of this model be formulated a likelihood. The MLE actually corresponds to the classic PCA solution. So this is a really I find this result fascinating because we apply PCA to all sorts of problems, in particular to data that aren't Gaussian distributed. But it turns out that you can derive the same solution under assumptions of Gaussianity. And for me, what that means is that you can define a model that's interpretable, but it also applies when you have violations of those assumptions. Okay, so the generalization that we propose, we have this x sub I k. Now k is going to index data set, so we can have 1 to capital K data sets. And then each data set can have a different number of features. So we're going to have a piece of K index there. And so the data from the ith subject in the kth data set, we're going to have our mean, but now we're going to have the z sub I, these latent scores. There's no index of K here. So that's going to be how we capture, that's going to be our data integration. That's going to be how we capture shared information. So this W sub JK matrix, that's going to be PK by R sub J. I didn't put just to avoid notational overload. I didn't write everything out, but this ZI matrix is going to be, say, dimension RJ. So these are going to be the loadings that are specific to each data set. So each data set gets its own loadings. And then we have the unique information, i.e. the individual information captured by these B sub IKs, which also get their loading matrices. So these B sub IKs are unique to each data set, while the ZIs are shared. Okay, and then we're going to assume within a data set isotropic noise structure. So we're going to have this low rank structure to simplify things to give us an interpretable model. And these E sub IKs are going to be ID, and they're going to get a data specific variance sigma sub K. So I guess let me just pause here. Are there any questions so far?
I'm not seeing any questions here, but just to clarify, I do have a quick question that at this point you're not introducing non-Gaussianity, right? You're just assuming that all
just one second.
are Gaussian.
I have a very different talks in the first part and second part. Second part's all about non-Gaussianity, and this one's all about assuming Gaussianity.
Mm-hmm. Yeah, and I think this is how you sort of separate or compartmentalize the issue of data reduction. This kind of approach, Projive lays down the baseline on how you approach data reduction.
Yeah,
I suppose.
Yeah, well, in some sense, but in another sense, well, so this next slide. So this model, a similar model was proposed, but it's also in some senses novel. So yeah, but it's more familiar because it's assuming Gaussianity versus when we move to the ICA world. So we were actually working on this for a while before I found this citation. So there's this old factor analysis paper. It's called inner battery factor analysis. So it turns out that there's an MLE that you can derive for this inter-battery factor analysis, and basically you have those ZIs. So going back here, the model is basically capturing these ZIs and ignoring, when it gets the estimate of WJK and ZI, it's ignoring that individual structure. But it hasn't, so yeah, there's this kind of gap because then these jive models came out and they're closely related to inter-battery factor analysis, but I haven't seen that connection made in any of the papers. I haven't seen this paper cited in any of the jive papers.
Yeah, it's a very interesting find.
Yeah, it's really, yeah, and it turns out actually this this inter-battery factor analysis is described in a paper called Bayesian canonical correlation analysis. And so these ideas are floating around in the literature, and hopefully this kind of helps to kind of tie some of them together. So this Bayesian CCA does include as a subversion this model. But they have a different way of estimating it, and they only get this approximate decomposition where they basically estimate a full factor analysis model with shrinkage, and then if the components are unique to the data set, they shrink towards zero. They're close to zero. So we're going to take a different estimation approach and estimate these matrices explicitly. I'm not going to say too much about the algorithm that we're going to use, but we can formulate a likelihood and then derive an EM algorithm and this algorithm will simultaneously estimate both the WJ matrices and the WI components, both the WJ and WI components. And really it boils down all to covariance estimation. So in a really broad sense, what we're doing here is covariance estimation under low rank structures. And then these cross terms, this is going to be that P sub 2 matrix by R. So this is going to be a rank R matrix. This is going to be R by P sub 1. And so we have a low rank matrix here that captures all the covariances between the two data sets. And the way that we approach this, we use an EM algorithm. So an interesting direction for future research is try to use some of the classical likelihood-based approaches, like likelihood ratio tests or BIC. But so far we found this permutation test that we do related to permuting the joint scores from two different sets and seeing if the correlation is significantly different from expected by chance. That seems to work pretty well. But we can talk about that more later. One of the drawbacks of working in this PCA world and the kind of factor analysis world is identifiability is challenging. And it turns out it's even a little bit more challenging for this particular model, that you could have this linear transformation of the joint loadings, and then multiply that by its transpose and inverse in the second data set, and it turns out this likelihood is invariant. It's you have to do a little bit of work to show that, because you have to redefine the individual noise components, but there's some cancellations that you get. When you have more than two data sets, some things cancel, and then we only have to worry about the special case where this is an orthogonal matrix. So under these orthogonal rotations, the likelihoods are invariant for k greater than 2. So when we're looking at these models, the kind of accuracy we're looking at, we'll be using subspace metrics, things that are invariant to these types of transformations. So I want to just kind of briefly touch upon the simulations. I'll just try to walk through the figure to give you an idea of how we looked at accuracy here. So we had n is equal to 1,000 subjects here. In the first data set, we had about 20 features. This will be similar to our AbbVie data set where we have like 22 cognition measures. In the second data set, we had 200 features, which is similar to the number of morphometric features we have in the AbbVie data set. And so in this first panel here, we simulated data following the model assumptions. So the ZIs were Gaussian distributed, and the B sub IKs and the noise, everything is Gaussian here. So this is kind of the best case scenario of what we expect. And we simulated it under different amounts of joint variance. So when R sub J1 is equal to 0.05, that's this panel here, and R sub J2 is equal to 0.05, that corresponds to this panel. So this is a very low joint signal. So this is the hardest scenario under the model assumptions that we evaluated. And so you can see here, this is our method. It's a little hard to see that that's orange, but our method is the furthest to the left. And you can see how we're outperforming these existing jive implementations. So I talked about a-jive. There's also this generalized PCA approach. That's it's really like a GLM version of PCA to use, but one of the cases that they have is a Gaussian distribution. And then this is the original R-jive algorithm from 2013. And I didn't tell you what that algorithm is, but it's an iterative algorithm that iterates between the joint and the individual subspaces based on using MSE as their criteria of accuracy. Okay, so the point is this orange thing is lower. When we move into the high signal scenario, I'm just going to jump to this. We do see some of these other methods perform pretty well. a-jive is doing pretty well here, but we're still doing a little better, in particular on these joint scores. Okay, so we see some improvement in the estimation in these simulations. If we looked at, so that was looking at the scores. We can also look at the loadings, which we get some more variable estimates for, but again, we do see this orange tends to be lower. When we get to this easier setting of estimation, we do see the methods do fairly similar on the loadings. It's kind of interesting. We don't have this correspondence between how well we're doing on the scores and the loadings. So just to remind you back here, so that you can have some methods that do well on one component but not the other. Okay.
A quick question here, Ben, about the measure that you're looking at, the chordal norm. If I recall, it looks at is it squares of the, what's the name? The skewness and the kurtosis? Is that the one? Or am I mixing things
No,
up?
that one. So that the chordal norm here is looking at the principal angles between the data sets, and then
Oh, okay, it's a different one.
yeah,
Got
so
it.
it's basically the inner product, and looking at the sine squares. So you can get those angles. They're related to the inner product, which is AB cosine theta. But yeah, that extension to the multiple to higher dimensions to subspaces. So it ends up being the square root of the sum of the sine of theta squared. Where the thetas are all the principal angles. So you have R, or those principal angles, when you have R joint components.
Thank you. And just double checking, panel B here is for the Z variables, right? The latent variables.
Sorry, say that again.
The panel B, is it for the Z variables
Yeah,
or
sorry.
the W
Panel B is
variables?
for the Z's.
Z's, isn't it?
Yeah.
Okay, got it.
Well, this is the Z's.
Mm-hmm.
These are the B's. So these are the individual scores. And then panel B are for, this is the WJ1, this is WJ2, this is WI1, WI2. Yeah, sorry I didn't explain that.
Thank you.
Okay, so that's when everything is Gaussian. But like I said, this PCA we apply much more generally. And we do see some, so in this case, we simulated a mixture of Gaussian scores with some pretty different, it's a mixture of 3 Gaussians, I think, with some, so it's a trimodal distribution here. And then the loadings, we made -1s and 1s, so we can just simulate that from a Rademacher distribution. Okay, so here we're violating the Gaussianity assumption. Mixture of Gaussians is highly non-Gaussian, even though it's a mixture. And we see that we still do, like, all these methods are pretty inaccurate with a very small signal, although we do technically do better. But then as things move up, you can see how we're still outperforming these other methods, even though these other methods aren't likelihood based. And even though we have the wrong likelihood, we still end up outperforming them in the simulations. So that was pretty cool. I kind of thought this would work out the other way. where AGI would maybe do better than us for when we're violating model assumptions. And similar patterns in the loadings. Okay, so the application that we're going to use here is to the Alzheimer's. It's actually the Tadpole challenge. I don't know if you all are familiar with that from like 2018. But what we're going to do, where the challenge was to predict what would be going on in a future release of the data. So here we're just going to use a single time point. And an extension of these approaches to longitudinal data is something we're working on. Or we'll be working on. We haven't started yet. But the single time point with 587 adults that have the full battery of cognition. So this is about 20. Yeah, well, this is the 22 variables. Include CDR-SB, the mini mental state examination, Montreal cognitive assessment, some of these measures of cognition. And the second data set we use are these derived free surfer measures. So we have cortical thickness, surface area, cortical volume using that desiccant parcellation. And along with some subcortical volumes and other structures, ends up being 245 variables that we have here. So an interesting discussion is, like, we could have potentially define 1, 2, 3, 4, 5, 6, 6 here, and then something like 5 data sets down here. We could have defined 11 data sets. We could have had capital K equal to 11. And that's a question that I'd like to look at more. But here, the idea is we want to treat, we're kind of aggregating the data in a sense to result in 2 data sets that we want to treat equally. So otherwise, you could have one modality kind of dominate all the others, or kind of the joint association between 2 modalities tend to dominate other things. And we really wanted to emphasize the cognition and morphometric connections rather than connections within morphometry. Okay, so skip that. There are some tests to try to derive the rank of the signal before you address the rank of the joint signal. So the rank of the signal includes both the joint and the individual structure. But really, we found scree plots is our preferred choice, because these permutation-based tests tend to select a huge number of components, and then we have issues with overfitting. Yeah, so we end up using scree plots to determine initial signal ranks, and then we have a permutation test for the joint rank. And we end up capturing about 21% of the variance in the cognition data set with just that one component, and then 10% of the information in the morphometry data set. So we're estimating, this is saying that 10% of the variance in morphometry can be explained by the cognition, which sounds big. And then the individual proportions of information. So this is just kind of what the dimension reduction is doing, how many of the directions we're throwing out as being attributed to noise. So we get 36% in the morphometry, and 50% of the information we retain. So these correspond to the same thing as PCA. Sometimes you'll see PCA use like a threshold of like retain 90% of the variance. So this is retain 50% of the variance. Oh no, excuse me, this is the individual proportion. So this is, sorry, I said that wrong. So it's, anyways, this is just the individual proportion is 50%, not the total signal. And this is the 36% for morphometry. So that's what's going on into the WIK, BIK portion of the model. Okay, so we look at the loadings here and see what pops out. So I have a picture of the brains in the next slide. So this MMSE, the mini mental state examination, Montreal cognitive assessment. So these point in the opposite directions as these other measures of cognition, which is appropriate. So so these ones, the higher I think means worse, and the other ones higher means better. I might have gotten that backwards, but anyways, they have different directions that you expect. And then we see how those we get 22 of these loadings, but we're just showing the 10 largest. And then on the other side, on the morphometry, we have 245 of those loadings, but we're just showing the top 10th percentile that are the largest in absolute value. And you can see some of the things that come out here, like the entorhinal. So that's one of the classic, or that is considered a potential biomarker of AD and MCI. Then also the hippocampus here. So that's also these regions that are pointing in the opposite direction. So it's actually ventricles. So in general, big ventricles means a lot of brain atrophy in this setting. So we expect those to point in the opposite direction. So that's also a really sensible result. And then we can plot some of those on the cortical surface. Some of those structures are subcortical, like I was talking about, the hippocampus. But then like entorhinal comes out here. So these are some of the cortical areas, fusiform. And then we have these temporal regions. So this is interesting. There are some forms of dementia associated with temporal issues. So anyways, this pops out. And we get this nice left-right correspondence. We're not giving the model much information at all. We're not telling it about left-right correspondence. So this is being learned in that joint structure. So that's cortical thickness. Remember we had cortical thickness, cortical volume, and also surface area. Surface area didn't really pop out in these joint loadings, incidentally, but the volume and thickness did. And so anyway, so again, the entorhinal comes out here in the loadings for cortical volume. Okay, so we get these scores from integrating cognition and the morphometric features, and then we can look at those scores and whether or not they relate to classic risk factors or to disease diagnosis or to other classic kind of biomarkers. So this is looking at the APOE4 count. Does that differ when we look at these joint scores? So we just have one joint score for each subject because our joint rank was equal to 1. We do see a trend, some separation in these different groups. These joint scores do have some separation. That the pseudo R-squared isn't super high, although pseudo R-squares tend to be lower than R-squared when you interpret them. But the T statistic is super significant, looking at whether or not these, or the F statistic, looking at whether or not these groups differ. And then looking at diagnosis, we also see a strong pattern. Some of those variables that are included in cognition are related to diagnosis, although allele count, the APOE4 was not used in diagnosis. So in some sense, if we weren't seeing this, we'd be worried. But this is something that we don't use any of this information in the model estimation. Okay, and then similarly, we don't use any of the information on PET that you can look at this data set, and we can look at whether or not cognition and these kind of cheap T1 markers are capturing some of the same information that we get in PET. And this is looking at the brain metabolism. So this is FDG PET, and we see a pretty decent R-squared, at least from my perspective, looking at these joint scores in FDG, and then also with the AV45 that looks at amyloid beta load. We see a pattern there that's significant, although the R-squared isn't quite as high as FDJ. Okay, so this is information that wasn't used in our data integration, but we're just from cognition and from the T1 image, we're capturing some important information as revealed by these other potential biomarkers. Okay. So I'll just quickly conclude the first part, and we'll just do a kind of quick version of the second part. But so we propose this probabilistic version of Jive, so with this latent variable to capture the shared information. And we saw in simulation some improvements in accuracy over the existing methods, and then we saw some nice relationships when we looked at the joint structure from cognition and dementia and extracted those scores. Those scores seem to be capturing some interesting biological information. Okay, so let's so that was the Gaussian part, and now we're going to launch into the simultaneous non-Gaussian component analysis called SING for data integration. So this is joint work with the co-first author on this paper, Dr. Irina Gainanova, who's an associate professor at Texas A&M. So this is a review of the slide I had last time, but this time we're going to look at data integration. We're going to be integrating the working memory task for the Human Connectome project with some resting state, just some static functional connectivity matrices. So the question we're asking are cognitive task regions. These ones involved in working memory. Are they related to spontaneous brain activity? So this working memory task, I think a lot of you are familiar with it, but this is a two-back task. We'll be using the two-back contrast here, and then also using this resting state fMRI where they're just staring at crosshairs. So, I mean, a lot of us work with resting state data, but it's kind of nice to step back and think that it's still an interesting biological question whether or not when someone's in the scanner looking at something like this versus something like this, are we seeing information, brain activity that is related? Or I think it's a very interesting question. So that's just whether or not there's joint structure between these two things is interesting. And then we're also going to be looking at whether or not the scores that we derive can be related to external variables. Related, in this case, we're going to use fluid intelligence. It's kind of a fun one to look at. So I mentioned this kind of in the Jive slide. It's actually the input in this whole thing is the same as in that 2017 jive paper using a jive. It's like the fourth slide I showed or something. So it's a z statistic from the 2-back versus 0-back contrast. So we get one of these z statistic maps for every subject, and that gets vectorized into about 60,000 locations for each subject. We have 1,000 subjects again. So that's going to be the X data set. We're just going to do 2 data sets. It's future work to integrate more. In the Y data set, we're going to have these resting state correlations. And we're going to use the multimodal parcellation from Glasser from the 2016. So it's about 360 cortical regions. We do also include the subcortical regions, but I'm not going to talk much about them in this talk. Okay, and then these modules that we're working with here, like we have visual and defaults in green, executive controls in red. So those come from this paper by Kiki and Abdallah, and those are what have formed these axes here. I'm going to skip over what each one of these means, but this is basically we just get a correlation matrix for each subject, and this correlation matrix we extract the lower diagonal, and then that gets vectorized and forms a row of the Y data matrix. Okay, so the lower diagonal list is input. So we're not telling the model anything about spatial location. We're also not telling it anything about the modular structure or anything like that. Okay, so this is going to be, it's going to end up being a really different approach to dimension reduction than some of the popular methods that are being used. So this is an overview of Join ICA. And the thing that's going to be common through all these is that they're basically using PCA for dimension reduction in some sense. And so in Join ICA, you concatenate the data sets, and then you do a PCA on the concatenated data sets. And that's what's doing the dimension reduction. The ICA in Join ICA is going to rotate that subspace, and that rotation improves interpretability. So the way this is also applies in general like group IC and stuff. We have to kind of switch dimensions to talk about that. But in general, the subspace is being chosen, the information is being chosen using PCA with ICA methods. Whenever you're doing pre-processing using PCA, that subspace is being chosen based on variance. Okay, and then ICA is just a method to improve interpretability from that perspective. And then there's also parallel ICA, which iterates between an ICA step and then maximizing the correlation of matched columns, and then multimodal CCA. So sometimes I see MCCA stand for multi-set. So we have more than 2 data sets, and sometimes it stands for multimodal. But some of the MCCA papers just have 2 data sets, which is the approach we're going to be adapting here. But that multimodal CCA, there's an initial PCA step that's done in these high dimensional settings. And then there's actually a CCA on PC scores. And that's actually, this is also a really cool connection. It turns out that's really similar to what Jive is doing, even though it formulates it all as equality of column spaces. Multimodal CCA, but then you get 2 separate, you get canonical variants, different ones for the different data sets. And then the Join ICA component of MCCA plus Join ICA is going to be, again, rotating those loadings to maximize non-Gaussianity. You concatenate the loadings. But it's that rotation similar. It's this step. It'll be doing this step where these V matrices are the concatenation of the 2 data sets. Okay, so the point here is that PCA is a crucial step in these. Okay, so we're going to try to take a different approach and see if we get anything meaningful. So the idea here is to use this information measured using 3rd and 4th moments instead of 2nd moments, i.e. variance is the 2nd moment. So here we're going to use skewness and kurtosis. And we're going to propose a matrix decomposition that looks similar to the Jive model, although there's some important distinctions here that this noise is actually it's not a full rank noise. So we get these different subspaces, but the information that's being shared is going to be captured by these M sub j. We have to kind of rotate our brains a little bit, because in the first half of the talk, this corresponded actually to the Z sub i's. So in the first talk, Z sub I was a first part of this talk, the Z sub I was a vector of length Rj. So now what this ends up being is all those n subjects, forming a matrix that's n by Rj, with each row corresponding to a subject. All right, so that's going to be capturing our joint structure. But then these S matrices, we're going to have a joint subspace and these individual subspaces. These S matrices are going to be non-Gaussian. They're going to have maximum non-Gaussianity under these constraints, where basically the constraint is the joint scores are equal and the individual scores are unequal. Okay, so this is the objective function that we propose, and let's see, I'm doing on time. Got about 10 minutes. All right, so it looks a little, so this pre-whitening thing is always a point of confusion when talking about ICA methods. But I'm just going to flash it up as being, we have these whitened data matrices X and Y, and we have to define it in a funky way, but this ends up just being the M, the columns of M. So going back here, M sub J, that's N by Rj. This ends up being one of the columns of M sub J. And initially we get a version that's unique, or that's different for each data set. Let me not call it unique, but like in canonical correlation, you get a canonical variable. So that's why we have, it's basically an MJX matrix and an MJY matrix. And what we do is we penalize the distance between those two to make them more similar. And so for a sufficiently large row, that corresponds to equal MJs. So that's the joint structure part. But then the dimension reduction step here is maximizing non-Gaussianity. So this is like an ICA within here, but it's actually a little different. It's actually a non-Gaussian component analysis, because we're using this for the dimension reduction. Rx is less than n, Ry is less than n. So we're only keeping these components that maximize non-Gaussianity, and components that are closer to Gaussianity, that are like kind of Gaussian, we assume those are meaningless and that those are removed. So this is achieving the dimension reduction. Let me just see, are there any questions about that?
Yeah, I think that was the measure I was talking about before. I got confused.
Yeah, that's the one. It's the Jarque-Barra test statistic.
Mm-hmm. Yeah, but I think this is a great point that you bring up, that this approach is actually doing data reduction. You're selecting the components that maximize independence from the full data directly, right? You're not doing a initial step to reduce based on variance. This is reducing based on non-Gaussianity. Whatever is most different from Gaussian, you keep in the data. Whatever is similar, it automatically is not included. It's in the data, but not in your projection.
Yeah, exactly. Yeah, so it ends up, so we're simulating this data, so you can simulate from a different model and make Join ICA and MCCA look better. So this is just when we simulate from this model, here I'm going to gloss over the details, but these are the joint components and the joint scores. These are the true components, and these are what we get with SING. They're like almost the same. You see they're almost the same. And this is what you get with Joint ICA. And the point here is that PCA step is retaining high directions of variance, and I can create toy examples where it removes all the variance. You control this in the simulation. you can make the non-Gaussian components correspond to directions of low variance, and then they get completely thrown out. So this is like a toy example where we see most of the information in the joint second component from this second data set, or I call this the first data set, second component, sorry, first data set, second component, data set X, gets thrown out. And you can see it still does kind of get some of the subject scores, but they're noisier. But then these loadings, we really lose that information. But yeah, you can construct examples where it completely fails.
I wonder if you consider the case where the direction accounts for high variability in one data set but not the other.
Yeah, we do. So we have simulations there, and that's actually what's going on here. Is the second component has more variability. So the PCA plus ICA does a little better. If you have a ton of high variation in one of the data sets, then that can swamp things and you still do well on the joint scores. But then we have some simulations examining that. In general,
I was wondering because if you're looking for a shared subspace, then if you have strong variability in one data set, it helps recover that direction from the data set with low variability. Explain.
Yeah, that's
in theory.
Yeah, that's the idea with some of this.
Mm-hmm.
So this is kind of an intermediate scenario where it is helping it a lot. If we were just to do a separate decomposition on this, we wouldn't get the joint component at all.
Mm-hmm.
But then it's still not doing a good job helping it get the loadings. So it's helping it get those joint scores, but not the loadings.
Got it.
Yeah. So we do have a lot of simulations that I didn't talk. I'm just gonna jump quickly to try to explain the data example, because I had no idea what would happen with this, because I mean, PCA is kind of the way PCA plus ICA is really how we define resting state networks. So what does the world look like when you don't use PCA? So you get these small patches of cortex, and initially I was like, well, they're so tiny. What does that mean? So this is in the task map. We get this. I mean, this is a couple hundred vertices or something like that. But they're really concentrated. So we get this cluster of super large loadings, and the rest is kind of background, closer to zero. So that's the task. Then if I look at the corresponding loadings of the resting state functional connectivity, only one node pops out. So it's just selected one node. We had about 360 of these nodes. And these are all from one node. So which node is that? We sum up the L1 norms, and we get one node pops out here. This is just looking at it. It's in executive control, central executive, so that's good because this is like cognition related stuff, working memory, central executive. Okay. But which node is it? This is what that multimodal parcel looks like. So we didn't tell it anything about the spatial correspondence, but it's actually finding that same. This is just selecting all the vertices that share joint structure with this resting state data, and you can see there's a strong spatial correspondence between it. So that's really cool. And if we look then at these actual, I can look at this row and look at how the loadings correspond for this node with all the other nodes in our resting state correlation matrix. And you see what this is connected with. These are parts of the working memory map. This is resting state data, but these look like working memory with this frontal region. And then this blue, that looks a lot like default. So we're getting opposite loadings in the working memory related regions and default. So that's super cool, because when I initially saw this, I was like, what is going on? Is this meaningful at all? But it takes a bit of work to find this kind of information. Let me jump to this one. So it turns out we get that spatial correspondence in most components. I think we got it in 24 out of 26. And so these are just the 5 other ones. And then if you look at, this is MCCA plus Join ICA, you sometimes get that nice spatial correspondence. This one's actually pretty good too. You can't tell, but this thing here is blue. These are sign ordered by their skewness to make their skewness positive, but then you sometimes get structure that seems to be totally out of line. So going back to whether or not these joint loadings are like capturing shared information, like these don't seem like shared structure if you're using this measure to get information about it. This thing's capturing some working memory components too, but it's not really capturing it in the corresponding resting state. So it's not clear we're really getting the joint structure here. Now I'm going to jump back to this just to show you what we get with the component in Sing versus Join ICA versus MCCA. So we get this much more nuanced, like smaller components basically of activation with Sing. This is the Join ICA component that's most related, that's kind of corresponding Join ICA component related. I didn't mention this is externally related to fluid intelligence. That's how I chose this component. There were 26 total that we estimated. Sorry, so of those 26 components, I was just focusing on the insight into one of them. So this is the Sing. This is that Join ICA kind of corresponding component. You see some large loadings in that region. And then, so that's the working memory one. This is the resting state one. You don't see much spatial correspondence. And same thing with MCC A plus JICA. We do see a little bit more highlighted in this region. I think it's like making some nice improvements over Join ICA, but we don't see kind of that strong overall spatial pattern. Okay, so I'm going to wrap up. I had a slide on, I didn't mention this because I kind of skipped over that. I do want to mention that the Sing model is hard to fit in the sense that this is not the Gaussian objective function, and you need a lot of initial values to fit it. So what I ended up doing was doing 200 initial values and selecting the one that maximized the objective function, and then doing it again in a second time, 200 initial values, selecting the one that maximized the objective function, and then only keeping components that were highly correlated between those two to try to improve the reproducibility. So that's one of the drawbacks when you go to this really non-Gaussian world, is the optimization becomes trickier. So to summarize, we have this new matrix decomposition for shared structure. It can improve estimation of the joint subject scores in our simulations, and then we seem to find this meaningful joint structure by this spatial correspondence between the different data sets, and we have some future directions for research to extend this to more than 2 data sets, and also see some models with different noise and potentially imposing different kinds of constraints in there, like to enforce sparsity, things like that. So I'll wrap up there. Just want to say thank you for listening. Thank you to the ADNI folks for making their data available. Thank you for the HCP folks for making their data available.
Thanks, Ben. This is great. I actually enjoyed very much the findings here. I was gonna before I ask a question, I was gonna check if anyone else here has a question that they would like to ask. You can let me know on chat or on the Q&A panel, or you can just raise your hand. There is an emoji at the bottom where you can click and select raise hand. Yeah, so I was going to ask about that observation at the end. You mentioned that you're basically fitting the model several times and selecting the model that minimizes the cost function the most. Mm-hmm. It's like you have an issue of multiple local minimum, and
right.
You're selecting the global out of multiple restarts. Do you choose? Is that the only difference, that you choose a different initial point?
Yeah, we're just initializing from different random matrices.
Mm-hmm. Do you have any constraints on that initial point? Like it has to the range of values or the rank of the matrix?
Yeah, so that's a good question. So here we have a joint rank test. It's again a permutation based test. So we do what we call a separate linear non-Gaussian component analysis on each data set. So it's basically the likelihood component analysis approach that I worked on during my dissertation. Okay, so we get separate estimates for that, and then we can estimate the joint rank, and then we take that as fixed in our that RJ is then held as fixed for all those restarts. So what we're changing are the different orthogonal matrices. So it's all in the whitened data. And the way we generate those orthogonal matrices, I've looked at in the ICA and the NGCA framework. And we didn't find much difference depending on how I just end up using simulating Gaussian data and then taking the SVD and then taking the singular vectors to get my orthogonal matrices.
Mm-hmm. Yeah, that's pretty standard.
Yeah, we've done stuff with like using the Latin hypercube sampling using the Givens parameterization of orthogonal matrices. And that was with ICA when we had square matrices, square orthogonal matrices. And it didn't help. So I just there's probably it's one of those things where you're like, well, maybe if I'd done this a little differently, it would have made a difference. But the point was, there we were trying to very systematically sample the different angles. So an angle is easy to think about partitioning that space from like 0 to pi with the identifiability. So it ends up just being a uniform distribution from 0 to pi. Yeah, I didn't find it improved. It didn't like lead to fewer restarts to find the global max. We looked at that a lot with ICA, and the first thing I worked on in imaging is a biometrics like 2014 paper on that, on the restarts. So it's like a problem in ICA, and then it's even a bigger problem in the NGCA world where you don't have the PCA dimension reduction first because the optimization space is even bigger.
Right. I'm wondering if I don't know if it would make any sense to think of it as more of a multi-objective problem, where you're optimizing separately the two not likelihoods, but the two cost functions for independence or non-Gaussianity, I should say. For each data set as separate dimensions, not in the same augmented function. If that would maybe give you a range of optimal solutions, if that would make sense to initialize, I'm thinking, your solution.
Yeah, for the initialization.
Yeah, before you optimize that they should share the same basis. Maybe find this Pareto front of solutions where you optimized. Basically, it's an optimization like the one you did, but with a regularizer that basically splits the weight from the proportion of weight of each term and the cost.
We kind of do that. So I didn't explain every step because there are a lot of steps, but. So I do those random matrices, but the input to seeing is actually not the random matrix. It's actually the solution to doing a separate ICA on each data set, to doing a separate NGCA on each data set, and then matching the columns. So that's a little bit. It's kind of like a warm start. But so from the whole thing will get initialized by the random, but then we have this step in between. So that we don't have to move that whole space using this curvilinear algorithm, and it's much faster that way.
I see. But yeah, I agree that this is computational effort is another concern here.
Yeah, we have a pretty fast algorithm for sparse ICA that maybe we can apply to this problem instead of and it basically uses it's basically using a Laplace distribution instead of skewness and kurtosis. That's one of the directions of future research. But it's something that yeah, it gives me a lot of consternation just trying to find the argmax of these actual non-convex functions.
Right, yes. We have a question here from Jean.
Oh, hi. I enjoy your talk. It's very good. Just trying to understand you present like two approaches. One is non-Gaussian, the other one previous one is Gaussian, with the similar framework of you have a joint component, you have your individual ones for each data set. I really enjoying this approach. Try to understand is when you're slide 22, you're presenting the loadings for the two data sets. Then there's one you mentioned for one data set of cognition, you have a less sets of loadings. For the brain data sets, you have more sets of loadings. That part I was confused, but I thought you joined. If that is a joint scores, then should two data sets have same number of sets of loadings? Is that the case?
Oh, so you're talking about this slide here?
The right, it says it's slide 22.
Oh, it's interesting. Okay, this one?
Right. You mentioned that for the cognitive scores, you have more sets of loadings, and then for the
Yeah. So this is, and so the number of the joint rank here was 1. So we only had, like, later on in the Sing stuff, we actually had 26. But when we're doing the jive, we just had one shared component, and then so each component we get one subject score, and then we get a loading for each variable. So in cognition, we actually get 22 loadings for that one component, but they all have this one component. And then for morphometry, we get 245 loadings.
that's because it's like more of the feature space, right? You have more of the score. Okay, but in terms of a joint, like rank is 1, so only have one component which is joint from the two data sets.
Exactly. So that's what's the same. Yeah.
Okay.
And the subjects is the same. So the number of subjects is the same. So that's the vector, of length n.
And another general question is, I see all you're presenting these cases, seems like you have more of subject number, like the one you have 500 subjects, but your feature space is less. It's like 300 brains or 20 or something for the cognitions. Have you tried because most of the cases we encountered is the other direction, which we have less component, but more variables, more feature space, and how this algorithm applicable in that condition?
Yeah. So if it's really high dimensional, you have to use the ajive approach, just for computational reasons. At least at this point, our EM algorithm doesn't scale to the 60,000 features like we had in the Sing approach. Okay. So in the second half of the talk, we had super high dimensional. But in theory, you can do it. The likelihood's well defined because of the isotropic noise. So that's actually the key assumption. Basically, usually when you have a covariance matrix, this covariance matrix, when you have p greater than n, is degenerate, and then the determinant blows up, and you can't do likelihood. So it does work here because this is well defined when we have the isotropic noise. But so in theory it works. In practice, it doesn't because it's too slow. Okay.
And then you mentioned that in the Sing approach that can handle much more high dimensional data. And then basically related to your answer regarding how do we find the K, right? The ranks. You said you did this individually test first, then you combine them to test again to see how many components is shared.
Yeah, so I didn't explain it here, but I can give it a shot. So we can do a separate non-Gaussian component analysis for each data set. So imagine you just get one M sub X matrix, and then you get one M sub Y matrix. So we do that separately on each data set. And then extract those scores from each. So we just look at those M matrices, and then we permute the rows in the second data set, and then calculate the maximum correlation between the permuted rows where the subjects are no longer in correspondence and the original data set. And so that'll give us a null distribution of the correlations that we expect just due to chance if there is no joint structure. So that's the test that we use. So we generate the null distribution of correlations under no joint structure, and then just keep the correlations that are significant.
Oh, that it will be decided how many you think that is common components. Okay.
Right.
Okay. Okay, that is one way to do it. That's good. Thank you.
You're welcome.
And the follow-up, that is for initializing your full model, right? This is the warm start you mentioned before.
Yeah, it does both things.
Mm-hmm.
Yeah. So it chooses both the RJ and gives us that warm start. Yeah.
Mm-hmm. That sense. Yeah.
Nice. Let me see if there are any more questions here. Okay, it looks like we don't have any more questions. I don't see raised hands. So we'll begin wrapping up. I'd like to thank you again, Dr. Risk, for this great talk. And I'd also like to thank everyone for attending today. We will continue this discussion later on Slack. So please check out the information that I put on chat here about how to get to our Slack channel. And with that, I will stop the recording. Thank you.
Thanks a lot.
And